Welcome back to Dnaofbioscience. So today's topic is about the Anfinsen's experiment. So we know that for protein folding, it is a physical process in which the polypeptide generally folds into its characteristics, or you can say the functional three-dimensional structure, right? So the folding of proteins into the complex three-dimensional structure is important for the proteins to carry out the vital functions of the body. So if there will be improper folding or misfolding, it will produce inactive protein. So the newly synthesized protein fold into the three-dimensional structure with the help of molecular chaperones, which are proteins. So the corrected folded proteins are transported to the Golgi complex, right? So Anfinsen in the year 1950, he clearly mentioned the protein folding by an experiment. Okay, so the process of protein folding was clearly understood by an experiment carried out by the C.B. Anfinsen in 1950. And this experiment provided the first evidence that the amino acid sequence of polypeptide chain contains all the information which is responsible for the folding of the chain into the native three-dimensional structure which is stable. Okay, so generally the Anfinsen experiment talks about the relationship between the amino acid sequence and the protein conformations. Okay, so let's get started with the experiment. So the classic work of C. Anfinsen in the year 1950 on the enzyme ribonuclease revealed the relations between the amino acid sequence of a protein and its conformation. Right? So ribonuclease is a single polypeptide chain consisting of 124 amino acid sequence and which are cross-linked by 4 disulfide bonds. Okay, so Anfinsen plan was to destroy the three-dimensional structure of the enzyme and to then determine what conditions were required to restore the condition. Okay, so he took two agents. First one is the urea that will disrupt the non-covalent bonds. Second one is the beta-mercaptoethanol. The disulfide bonds can be cleaved reversibly by reducing them with a reagent such as beta-mercaptoethanol. So in the presence of a large excess of beta-mercaptoethanol, a protein is produced in which the disulfides or the cysteines are fully converted into the sulfohydryls. Okay. So first first point is when he took first the protein called ribonuclease. So as I said, ribonuclease is made up of 124 amino acids and it cleaves generally RNA. So this protein, that is ribonuclease, it can be denatured by adding some chemicals or by heat. Okay, so let's see the process. First he took the ribonuclease, he treated with beta-mercaptoethanol in 8 urea. The product was a fully reduced, randomly coiled polypeptide chain which is devoid of enzymatic activity. So in other words, you can see the ribonuclease was denatured by this treatment. Okay. So in the first case, when you are using mercaptoethanol in 8 urea, the protein denatured. Okay. Now let's see the second step. In the second step, Anfinsen then made the critical observation that the denatured ribonuclease freed of urea and beta-mercaptoethanol by dialysis slowly regained the enzymatic activity. So when you are removing the beta-mercaptoethanol and the urea by dialysis, there will be slowly regain of the enzymatic activity. So when you first remove the mercaptoethanol, the sulfohydryl groups of the denatured enzyme became oxidized by air and the enzyme spontaneously refolded into a catalytically active form. And the detailed studies then showed that nearly all the original enzymatic activity was regained if the sulfur-hydryl groups were oxidized under suitable conditions. Okay. Then the third step is a quite different result was obtained when the reduced ribonuclease was reoxidized while it was in 8M urea, and the preparation was then dialyzed to remove the urea. Ribonuclease reoxidized in this way are only 1% of the enzymatic activity of the native form. So the reason is that the wrong disulfide formed pairs. So when you are removing the mercaptoethanol but still the urea is present, then there will be also inactive RNH formation with randomly formed disulfide bond. So the wrong disulfide bond formed. There are 105 different ways of pairing 8 cysteine molecules to form 4 disulfide. Only one of these combinations is enzymatically active. The 104 wrong pairing have been termed as scrambled ribonuclease. Okay. So when you are removing the mercaptoethanol, that is reoxidation takes place. Okay, but it was still in 8M urea. Okay. So in that condition also there will be random formed disulfide bond and it leads to inactive protein. Okay. So in the fourth step, the Anfinsen found that the scrambled ribonuclease spontaneously converted into fully active or the native ribonuclease when trace amounts of beta-mercaptoethanol were added to an aqueous solution of the protein. So when you are adding beta-mercaptoethanol, trace of that, then the renaturation process takes place. Okay. So the added beta-mercaptoethanol catalyzed the rearrangement of disulfide pairings until the native structure was regained in about 10 hours. And this process was driven by the decrease in free energy as the scrambled conformations were converted into the stable native conformation of the enzyme. So the native disulfide pairing of ribonuclease thus contribute to the stabilization of the thermodynamically preferred structure. So in this diagram, this is the native structure and this is the scrambled structure. When we'll remove the mercaptoethanol, but when there is urea, then the randomly formed disulfide bond and it will inactive the RNAs. So here the conclusion is the native form of protein is thermodynamically more stable. So when we are adding both mercaptoethanol and urea, you can see the denatured protein. Then when you are removing the mercaptoethanol but there are still urea, there will be scrambled spaces, that is the randomly formed disulfide bonds formed. Okay, and when you remove urea and introduce small traces of mercaptoethanol, then there will be formation of the native structure. So here is one question: the treatment of RNAs with 8M urea but without adding mercaptoethanol would also denature the enzyme. I already told there will be inactive ribonuclease with randomly formed disulfide bonds. Okay. So the conclusion is the disulfide breezes break and the atrophic forces between the amino acids disappear. Okay. So the enzyme is denatured into the tangled or scrambled form. The denatured process is then again reversing back by removing the denaturing condition, like removing the urea or the mercaptoethanol, or by lowering the temperature. So then the ribonuclease falls back into its native state when the denaturing conditions are reversed. So the native form of protein is thermodynamically more stable. So you need to remember 4 cases when you are adding mercaptoethanol in urea. Then there will be denaturation. When you are removing, then there will be renaturation as the oxidation of sulfhydryl group takes place. Then when you are removing the mercaptoethanol but still there is urea, then the inactive RNAs will form with randomly formed disulfide bonds. Then when you remove the urea plus there is mercaptoethanol, traces of mercaptoethanol, then it will renaturate the protein and it will form the native conformation. So this is all about the Anfinsen experiment. This is the relationship between the amino acid sequence and about the protein conformation. Thank you very much. If you like this video, hit the like button, share with your friends, and do not forget to subscribe my channel. Thank you.